Welcome back to my channel. Today we'll be testing our aspirin. The aspirin we prepared previous video. Today we just have to test it. There are two ways to test aspirin. One is through melting point. The other one is by using ferric chloride. And so we are not going to use melting point to test for our aspirin, but we are going to use ferric fluoride. Ferric fluoride react with phenols present in salicylic acid, and so we are not going to test purity per se, but we're going to test the impurities that are present in our sample to know how pure our sample is. Let's head down to the experiment. So this is the experiment. Let's conduct the experiment. Permit me to use my reference. It's just a simple experiment. It doesn't really involve hazardous chemicals. So permit me to use my office. For your reagent, you have methanol. And why the choice of methanol? Methanol, helps in dissolving the samples without interfering in the results, or without interfering in the reaction. You have ferric chloride, and this is 10%. Ferric chloride reacts with phenol, and it helps to identify the presence of phenol that is present in salicylic acid, and we can detect that by a color change. It can be a violet color or purple color. So with that, you can know that there is a presence of phenol in your sample. Remember, we are testing for the impurity. And the impurity we're talking about here is the major ingredient of this aspirin, which is salicylic acid. And salicylic acid contains phenols. For samples, you have your salicylic acid, and you have your aspirin. This aspirin is the one you have already synthesized in the lab, and so that's your sample. Then for your apparatus, it is not every of these you can see here. You have your conical flask, you have your beakers, you have your measuring cylinder, you have your pipette, you have your funnel, you have your spatula, you have your two test tubes that are kind of label them to identify, for easy identification. Then you have your test tube rack, and I have my filter paper. This is just here it's just a temporal, filter paper. I don't really need it. Alright, so what is the procedure on how to test this aspirin? Number one, you have to take a little quantity of aspirin, and as I labeled here, you take a little quantity of your aspirin, a little quantity. You don't need to measure this, you just have to take a little quantity of aspirin into your test tube. And then you keep that. Then you take a little quantity also. You can also clean your spatula. You take a little quantity of your salicylic acid into to your test tube. Label salicylic acid. A little quantity. Okay. And after that, you have to measure 2 mils of methanol. Methanol will help to dissolve our solvent. 2 mils of methanol. You can measure it direct, or you can use your pipette to measure. 2 mils of methanol, and add to each of your samples 2 mils. Make sure that it is 2 mils. Don't get it more, don't get it less. Then you add this to, I'm adding now to aspirin, 2 mils. Then you can agitate this, and allow it to rest while you measure another 2 mils of methanol. The pipette is also calibrated. If you want to use pipette to measure, also acceptable. Measure 2 mils. Then you add your 2 mils in salicylic acid, 2 mils of methanol. As I said earlier on, we choose methanol because it will not interfere in our reaction. So let's test for the impurities present in both of these samples. This is salicylic acid. You just have to take a drop of your ferric chloride and just add a drop of ferric chloride. You can see the color change. This is purple or violet or so. I believe it's purple. If you shake this, the color remains. It doesn't change. It remains. You know, this is because ferric chloride forms a long chain ligand with salicylic acid there, and that is the reason why the color is not changing. And it tells you that there is a presence of phenol. There's a presence of phenol in this reagent, in this sample rather. Then coming to your aspirin, you just have to add a drop of your ferric chloride. You see, the color disappeared. There was the presence of purple, but the color disappeared. This is because the color change informs you that there is a presence of phenol there, but the disappearing of the color not being permanent is telling you that, you know, the phenol was exhausted, like it wasn't permanent, it was exhausted. So this simply means that, you know, all the phenols or salicylic acid present in your sample, you know, was kind of exhausted. And if you compare these two together, you see that this color remains permanent, while this is, you know, kind of light. So this tells you that your sample, you know, is kind of pure in a way, while this is not pure. So this contains a lesser or no amount of salicylic acid, while this has a very more, you know, amount of salicylic acid. Now remember also that this purity test is not a standard purity test of, you know, of pharmaceutical drugs, but just a laboratory test. And so in no account will you think that this is pure and you take any of the, you know, the synthesized aspirin as drug. You know, this is just for your knowledge. Thank you for watching, and if this is your first time of coming across my page, click on the subscribe button. See you in my next video.